Hello, everyone, and welcome to today's webinar titled Improving LCMS Workflow Efficiencies with Batch Queue Sample Submission and Automated Data Analysis. I'm Susie Valdez of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is sponsored by Agilent. So let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you'd like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, click on that Ask a Question box located on the far left of your screen. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located in the top right corner of your presentation window and follow the process to obtain your credits. Without further ado, I now present today's speaker, Ryan Rademacher, Clinical LCMS Product Manager at Agilent Technologies. For a complete biography on Ryan, please visit the Biography tab at the top of your screen. Ryan, welcome. You may now begin your presentation.
Hello, thank you for the introduction. As mentioned, this is the Improving LCMS Workflow Efficiencies with Batch Queue Sample Submission and Automated Data Analysis webinar. As she previously mentioned, my name is Ryan Rademacher. Just a couple of quick words about myself. I am the Clinical LCMS Product Manager for Agilent Technologies. I've been with Agilent for about 8 years working in LCMS in one way, shape, and form. And as she mentioned, I came prior to that from the pharmaceutical industry as an instrumentation specialist. For today's agenda, we'd like to go ahead and first start off talking about LCMS analysis. And I like to talk about that in the context of a 5-step workflow. Each of those steps will include the sample prep, the chromatographic separation, the data analysis, data interpretation, and reporting. I also like to think about that same 5-step workflow as it goes over a 3-phase life cycle, specifically the method development, method validation, and the routine analysis phases of the LCMS analysis workflow. Following that, I'd like to talk about optimizing LCMS for routine analysis and looking at that through the lens of a statistical complexity or modeling it as a statistical complexity and where the practical applications of that model come into play, specifically utilizing our Agilent Technology Study Manager software, which has features to improve your workflow and a demonstration as well. So that's the agenda. Finally, we'd also like to have question and answer available for you as well. So the LCMS analysis can really be looked at as having 5 steps, as we talked about in the agenda. And each of these steps change a little bit during the different phases. So during the method development, you can actually see sample preparation. And sample preparation being the first step actually is quite an elongated process. It takes up a lot of time. And that sort of makes sense because during the method development phase, a developer would be tasked with a lot of questions, particularly around the sample preparation being a new assay. Some of those questions could be, what's the ideal sample? Is it going to be coming from oral fluids? Is it going to be from urine, crushed human plasma, so forth and so on? Additionally, they would be looking at what type of workup would be required for that particular sample. And also they would be investigating whether that particular workup could be automated. So that elongates the sample prep step of the method development phase. Equally as long is the chromatographic separation. And again, this makes sense because being a new assay, a lot of time is going to be allocated from the developer into the chromatographic separation because it's relatively new. A lot of questions would come up, such as what type of column or mobile phase are required for this, how much sample volume is required to inject to meet the LOD, LOQ, and what are some of the MS parameters. All of that would have to be investigated and determined experimentally, which would result in an elongated step. Next, the developer would be tasked with determining the data analysis for the new method. For example, how are they going to quantitate or qualify the results that are generated? And also important would be looking at the data interpretation. Specifically, they would be looking at the data interpretation from the vantage point of what can the results tell them. And finally, they would need to develop a comprehensive report to capture all of the findings of the sample prep, chromatographic separation, data analysis, and data interpretation. And you can see that each of these steps is actually quite long, which makes sense in the method development phase. But the next phase that they would be looking at it is the method validation phase. And in this particular phase, you can see that the first two steps, the sample prep and the chromatographic separation, are much shorter than in the development phase. And that's because the results of the development phase have led towards a shortened period, more appropriately an optimized separation methodology, as well as a chromatographic separation methodology. So you can see that those are much shorter. However, any of the time that's gained or the time to results that's gained in the reduced sample preparation and chromatographic separation steps is usually lost in the data analysis and the data interpretation steps during the validation phase. And the data interpretation specifically is elongated here because the validator is really looking at what do the results tell them, and is there potentially any need to go back and change the data analysis previously. So you can see those two steps really cut back on any of the time savings that are gained in the reduced sample prep and chromatographic separation. Again, similar to the development phase, the validation phase requires a comprehensive report that's equal in length, but different in what it's actually conveying there to really fully articulate the findings of that. Now, the final phase, of course, that we're looking at today is the routine analysis phase. And you can see that in this particular phase, each one of the 5 steps has been greatly reduced. And that generally makes sense because routine analysis phase is all about repeating the same process over and over again. So you can actually see where it's been reduced. And we can see that each one of the steps has been ideally optimized, and the analysis is now fit for a specific purpose. As I mentioned during a routine analysis, really the lowest time to results and the maximum throughput are really the kind of the name of the game in this particular sense there too. Now, the previous examples have demonstrated that there's an elongated time during the earlier phases and can similarly represent the amount of extensive expertise that's required. As the technique moves into the routine phase, however, less expertise is required, but in order to continue to gain efficiency, specifically time to results and being able to use the time to results, it becomes necessary to employ other tools in order to continue to improve that efficiency there too. And in order to do that, I like to think about modeling how to actually continue to gain those improved efficiencies in routine analysis by using the statistical complexity model. For those of you who aren't familiar, statistical complexities are a section of mathematics that's dedicated towards probability modeling and determining what the complexity of individual systems are. And for this presentation, you can think of complexities as kind of the arch enemy, if you will, of efficiency and time to results. Those things are generally inhibited by complexities there too, or maximizing those things are. But complexities have a lot of applications there too, and they're often used to model probability, as I mentioned prior to, in machine-based learning. They also have been found to, they are often used to reduce conditional training in automated learning. So for example, in things like autocorrect and predictive script algorithms. But they also have anthropological learning capability applications as well, and they've demonstrated that they can actually be used to determine training for humans as well there. So they really fit the model. And it seems appropriate to apply these to how we can actually reduce the complexity or model the complexity inside of our routine analysis. The complexity that we're using today is coincidentally named the Rademacher complexity. I didn't write it, full disclosure, but it is one that nevertheless works for today's demonstration quite well, actually. The math on this is a little bit intimidating, or it can be a little bit intimidating. It certainly was for me when I first started looking at it. But essentially, what it's saying is you can model the overall complexity of a system by determining what the summed average of randomly generated sampling points are and their closeness to the elements of the system. So if you were to imagine randomly selected sampling points trying to figure out what the complexity are of a system over the entire system, you were to come up with the sum average. The complexity could be expressed as the closeness or of the average of all of those sampling points to the inflection points of the system or the elements of the system more broadly. So that's a mouthful. What does it really look like? Or how can we apply this to it there? So let's imagine that we have each of the steps in our routine analysis for a single cycle and where they would exist as moments on the 12-hour clock for each one of the phase. So you can see in our illustration here, we have a development clock, a validation clock, and a routine analysis clock there too. And we take each one of the steps, more appropriately the inflections between the steps, and we start to plot them. So we can see that in the development phase, we would finish up our sample prep at, let's say, 2 o'clock. The validation in the routine phase, we would say, oh, we would finish up our sample prep at 1 o'clock because remember, we optimized those based off of the results from the development phase. So now you can see that in the development phase, the chromatographic separation happens at about 4:30. With the validation in the routine phase, you can see that those are again at about 2 o'clock because we remember that we optimized based off of the development. Similarly, as we go into the data analysis, we can assume that in the development phase, it happens at around 7 o'clock. In the validation phase, this happens at about 4:30. In the routine phase, this would happen at 3 o'clock, though, because we've optimized both off of the development phase and the validation phase. And with the data interpretation, we can assume that for development and for validation, this happens at 10 o'clock, whereas with the routine phase, this happens at about 4 o'clock. And finally, with the development and validation, we assume that we finish that up at around 12 o'clock or midnight, whereas with the routine, we would finish this at about, let's say, 5:30. So I like to actually model these because it gives us different inflection points on the face of a clock and where they would actually fit into that particular clock there. So let's plot the transitions between each of these steps. So let's remove the face, the clock face, and let's look at the transitions. Now you'll notice that we're only seeing 4 transitions in each one of these phases. And that's because we're assuming basically infinite rest before we go ahead and actually start our sample preparation, and we're assuming that we have infinite time to look at our report once it's been completed there too. So you can think about this, if you will, as like I was born ready to do sample preparation and I died with a report in my hand, if you will, something along those lines. And we're able to do that because we're really only looking at the inflection points between each one of them, and that helps us with this model. We could potentially expand the model and change it a little bit, but for simplicity, this works quite well for the example. Now, where it becomes very interesting is when we actually start looking at how complex our system is. If you remember, the Rademacher complexity we talked about being the average of all the sample points and how close they are to the elements of the system, and I told you that in this particular side, we're looking at the inflection points as representing points of complexity in each one of our models. And so we've randomly selected a sample point, which I've designated as C1, and it's sitting at about the 9 o'clock position. And we're looking at that as the largest dot or largest possible dot product, or more appropriately, the most acute angle that exists to any of the existing transitions that we have in there as well, which are A1s, which I've assigned as A1. So you can see that at the 9 o'clock position, and again, this is just a single sample that we have here, the development and the validation phases show tremendous complexity relative to the routine analysis phase. And that's because the C1 at the 9 o'clock position for both the development and the validation is incredibly close to In this particular case, it's close to the data interpretation transition that we're looking at there too. Whereas in the routine side, it's quite a ways away from all of the transitions of the inflection points that we talked about. So you can see that the complexity at this point is proportional to the value of the dot product, or the max value that you can have. And you can also see that while the development phase and the validation phase are relatively equal, they're vastly greater than the routine phase there too. And the reason that we see this, and now we know that naturally a single measurement does not make a really good model, and statistics are really in general. And the best model is going to include the average of every possible random point. So you can imagine if we started sampling at 1 o'clock, 2 o'clock, 3 o'clock, 4 o'clock, so forth and so on, we would get better and better models. But still, it's not difficult to see just visually that in the routine phase, because of the abundant free time that exists in the latter half of the clock, that it's the overall least complex model. And I've demonstrated that with, just coloring in the green crayon here too, that demonstrates all of the free time that we have in the routine analysis there too. But the complexity of the routine phase is undoubtedly the lowest overall for the full time range. But as I mentioned, a single data point doesn't represent a very good model. So let's consider it at another point. So for example, if we took it at the 3 o'clock position, consider it when we're at the 3 o'clock position as we've done here. And in this particular case, we can actually see that the development and the validation, at least at this one data point, are the least complex of both of them. So at this particular point in the routine phase is actually the more complex model there too. Now, why do we bring this up? And it's because it demonstrates an interesting phenomena that exists in statistical modeling. That's one of the powerful tools for the Rademacher complexity is that it actually shows that complexity can be localized and concentrated into certain areas. And while those certain areas would generally be more complex, it's going to result in the minimal time to results workflow. It actually can become less complex overall. And one of the things that's really interesting is when you consolidate all of your complexity down to a very, very brief period of time, you can actually have an overall less complex model, even though you have more transitions. So if you can imagine, you could actually have more transitions, but if you consolidate them into a certain area, if you localize them, it actually statistically is less complex there too. So it's kind of an interesting observation that you get from the statistical models that wouldn't intuitively come out. However, the problem is that it's not very likely. So one of the major issues with this is, although you can localize your complexity into a brief period of time, and overall, if we assume that it's a 12-hour period, it's far less complex, right? That's a good thing. The downside is it's not very realistic. As I mentioned before, the efficiency and time to results are the entire reason why you would do a routine analysis. And so it doesn't make any sense that you would simply run one cycle and then wait until the 12-hour period is over before you would start again. No, that's not very likely at all. So what's more likely is what you would see in the illustration here, where you've started one cycle and you've actually increased that one cycle because you've started another cycle there too, because you know that you can complete this within a 12-hour period, and that's far more likely. You'll also notice that there's an additional inflection point that's been included here. So before we only had 4 inflection points. Now for our second cycle, we actually have 5. And the reason is that we have 5 is because we can no longer assume that we have infinite rest before and after. Previously, we had kind of used that analogy of, I was born ready to do sample prep and I died with a report in my hand. Not so in this case, because at some point we have to transition from the first cycle into the second cycle, and that requires another transition point there too. So you can see that the complexity, at least statistically, has more than doubled just by adding another cycle to it there too. So we've taken what was normally a far less complex cycle overall, and we've actually, just by increasing it with one additional cycle, we've more than doubled the complexity there too over our 12-hour clock. So that's a pretty abstract concept, and statistically it does demonstrate some interesting things about complexity in a routine workflow. But how do we apply the abstract concept of our complexity to a routine workflow? Now, within Agilent Technologies, we use Study Manager software, but more broadly, it talks about the complexity demonstrates that by combining the steps for an operation that's going to be frequently completed, we can actually reduce the complexity overall. So imagine if we take the amount of steps that we had. So we looked at 4 different transitions, of course, going from sample prep sample prep being outside there, but looking at chemical separation, going into data analysis, data interpretation, and finally reporting. If we're able to combine those into a single step, we can actually greatly reduce the complexity. So again, we've looked at 4 transitions. Now we can take that down to 1 transition. And our model, which looked like this off to the left-hand side initially, has now been reduced down to this. And again, we've received the same 2 cycles, but our statistical complexity has greatly reduced down because now we're looking at a far less complex model there too. So we do this by doing automated data analysis, of course, but there's other benefits to this as well. We know that combining separation and automated data analysis and reporting into a single step can greatly reduce the complexity. We've gone ahead and we've modeled that. And I think that's generally pretty intuitive too, but sometimes it's good to see the model as well to really walk through it. But it can also reduce the amount of screen time that's required and the training requirements. If you remember initially I talked about statistical complexities are often used in learning capabilities. Well, this is the epitome of that, right? So we've gone ahead and we've taken our staff and we've taken them from having to learn 4 different activities or 4 different transitions, chromatographic separation, data analysis, data interpretation, and reporting. If we've limited that down to just a single step, we've greatly reduced the amount that we have to train them on too. So it really is a very powerful tool. And of course, it's going to reduce the screen time as well because our staff isn't going to have to spend as much time in front of it doing all of those activities because we've reduced it down. The ability to schedule sample submission, if we're to combine that, for example, into batch Q submissions, around the instrument availability combines with this to further improve the utilization of the instrument and, as I mentioned, when it's coupled with the data analysis and reporting, is going to significantly reduce the time to results there too. And I like to demonstrate that with a standard submission. So if you can consider what we talked about before where we have 2 individuals, 2 technologists, medical technologists who would be submitting samples. You can see at the top there, the one would go ahead, the first one on the top would go ahead and finish the sample prep and would immediately start doing the chromatographic separation, followed by the data analysis, data interpretation, reporting. In the meantime, the second individual on the bottom would have all the way up until the chromatographic separation is completed before they had to actually complete and go ahead and start the chromatographic separation. So they would have to stagger this a little bit. And actually this actually represents the best case scenario there too. This is a situation where the individual that's on top would actually be able to do their data analysis, interpretation, and reporting offline. That's a very common workflow. We produce products, additional copies of data analysis software so that people can do that. However, there may be a worse situation where the single workstation, which is used for acquisition, chromatographic separation, is the same workstation that's actually utilized for the data analysis, interpretation, and reporting. And in that particular case, you can see how the sample submission has been greatly elongated and the time to results have really suffered for that too. So that's the worst case scenario. So then enter in batch Q sample submission. One of the things I talked about is with batch Q sample submission, really what you're doing is taking the ability to go ahead and kind of take up to 3 different or as many batches as you'd like to go ahead and proceed forward. And you could do all the sample prep at the same time and go ahead and submit. And really, it works around the instrument because as soon as one particular batch would be completed, the next one would be able to go ahead and start. So we're no longer staggering everything in that worst case scenario, and we're actually better off than that standard submission as well there too. So it's a powerful way to kind of combine that. As I mentioned, within Agilent, we do this with a software program called Study Manager. Study Manager is a software program that's included in all Agilent LC-MS triple quad systems, and it's designed to limit the software interaction and increase the throughput for routine applications. The nice part about this is, as I mentioned, it's included in all triple quads, and it has been for a while. So if you actually have a system that you're using, a triple quad system from Agilent, chances are you already have this in there. It's free. It's included with the software. You don't have to go through a third-party vendor to get it, and you don't have to actually, it's not add-on software as well. So you already have it. It's already free. It's included. And it does all of the things that we talked about in our statistical modeling and our batch queue submission example as well. It does combine our Mass Hunter acquisition, data analysis, and reporting software into a single software program, which permits a user to conveniently select from pre-configured methods from a drop-down menu. And it fits really well for the routine analysis because in a routine analysis, it's assumed that you will be doing the same thing or similar things over and over and over again as we demonstrated before. So because we're doing that, we can actually increase our throughput with the Study Manager tool by having drop-downs of specific methods that we know that we're going to be running. It also has the ability to automatically do data analysis and post-acquisition data analysis and automatically generate reports for our submissions. As I mentioned before, it combines with batch queue submissions so that multiple users can utilize a single workstation. You can imagine a situation where users would go ahead, they would prep their samples, they would set the tray into the instrument, run through the Study Manager workflow, walk away because we're doing automated data analysis and reporting, and their colleague would be able to walk up shortly afterwards with their prep samples, submit their samples as well into the Study Manager software and walk away. And as soon as the first one finishes, the second one would start. And that increases their labor utilization rate because they'd be able to do other things there too. Again, it allows the entire sample sets to be submitted for acquisition prior to the completion of other batches. So that's where batch queue submission really fits in as well. Some other features it has is the ability to do work list and work list import workflows. So the Study Manager software permits a user to submit sample sets from existing Mass Hunter work lists. If you can think about the work list that would have been created perhaps during the validation period or maybe even the development period, if those are already in place in your lab on the workstation, the routine analysis or the routine analyst would be able to select those work lists from the drop-down and be able to submit them. Or if you have an import file, so for example, from a spreadsheet, which could come from an LIS or a LIMS system there too, they would be able to import those samples as well using a drop-down map file, which would then translate all the files into a work list there too. And that would all be set up so that the routine analyst would, again, just have to do simple drop-downs and move through. The entire software program is really designed around improving the productivity and limiting the steps from sample submission to reporting. Finally, one of the other features that it has is the ability to import and export data to an LIS and a LIMS system. So as you can see in the illustration off to the right there on the screen, not only are you able to pull work lists, for example, from an LIS, but you'd actually be able to submit it, analyze it, report it, and then upload those results back up into your LIS there too, into a report that's specific for the type of assay that you're running there as well. So it can be configured to both export and upload the results into the LIS system as well as import them. So it really makes for a very complete full circle workflow and able to work through. So those are the benefits. I like to walk through before we go into the question and answer and just do a quick demonstration. Now, these are actually screenshots, obviously, from the system. I can't do a webinar with a live system, but this was all done on a system as well, so it's effectively a live demonstration. And what you're looking at right here is a shot of a workstation that we have. The Study Manager program, which again is included in all of the Agilent LCMS triple quads, has been set up and configured as an icon, and a routine analyst or a medical technologist would be able to simply double click on the icon to launch the Study Manager software and would simply select a study from that as well. As I mentioned, we talk about work list and work list import types of studies. There are a few others, but they're a little bit outside of the scope of this as well. That said, your application engineer or your representative would be able to walk you through those as well and give you some more details. But again, here in this example, we're just going to do a simple work list only selection. Again, you would select the appropriate file and work list from the drop downs that are on the right. This particular work list is called a demo work list. You can put some specific information for traceability. So for example, if you wanted to label the sample set with a lot number, or if you wanted to have some information about the submitter, I've used the generic operator here, but you could actually have some sort of unique identifier for your staff so that you would be able to tell who was submitted the study and what specific lots they had, or any information that would be relevant. That could all be included in this particular page. And then on the next, following the wizard along, just by clicking the next button, the individual would be able to select whether they want to do automated data analysis, and they would select from a drop-down of quantitative analysis methods and a drop-down of specific reporting options as well. And again, this could be reporting options that either create a report that pops up on a screen, or it gets sent out onto a network somewhere, or is incorporated into an LIS. That could all be configured as well. The final step would be just to verify the work list, making sure that all of the sample positions are correct and that the methods that are being used are correct as well. It's a sanity check, a verification, before finally hitting finish, and you would be able to submit the batch. So from here, you would be able to just go ahead and click you can see that the batch has been added to the queue, and the individual would, in this particular case, they would have to hit the start button, and that's because it's been sitting idle. But a colleague of yours could actually come and walk through that same wizard that we went through previously and hit submit, and wouldn't have to actually hit start. That's another feature that's included there too. You can configure it so that it actually goes into, if it's sitting idle for a long period of time, it can actually go into a standby position. But assuming that you have a high throughput workflow, your colleague would just be able to submit the batch. And then from there, you would just simply wait for it. Go into the lab, work on other things. Your colleagues could again submit other batches if they wanted to, until finally a report would display or save to a predetermined location, including an LIS, if you were. And that's relatively simple. Again, this is basically our software solution to reduce the complexity of your system and ultimately increase your throughput. So it's a very powerful tool when it's used. It largely just takes those additional steps that are associated with your routine analysis out, so that you're limiting the screen time, and potentially can reduce the training that's available for it. And also allow for batch queue submissions there too, so that you can really increase the productivity and efficiency in your lab. At this point, I'd like to go ahead and open it up to any questions, and see what we can help.
Thank you, Ryan, for that informative presentation. And we will now start the Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Simply click on the Ask a Question box located on the far left of your screen. So let's just get started. We have quite a lot of questions coming in already from our audience. Thank you for these questions. Our first question, Ryan, is the study manager available on the IVD product in the booth?
Yeah, actually, yeah. So for those that haven't seen it, we did do a launch, or we have a promotion of our newest IVD product, our K6460S. And being a triple quad, it is actually it does include the study manager. And actually, even better than that, right now, as a thank you to all of the clinical diagnostic workers there too. We're actually setting up a promotion on that particular product as well. So in addition to having the study manager capabilities, we're offering actually 3 years of extended warranty on that at point of initial sale for no additional cost. The details of that would be available, of course, through your Agilent representative. But yeah, we're actually promoting that, and it does have the study manager. So yeah, good opportunity to kind of plug the K6460S. So good question there. Yeah.
Thank you. And we have a related question. How much does it cost?
Oh, well, it's free. I don't usually get to say that in these when I do webinars. Of course, it's always the cost, and I have to say, well, talk to your representative, and they'll work with you. This is one of the few webinars I get to actually say it is actually free. It comes with the instrument. So the triple quads all have study manager included in it in the IVD product, so there's no additional cost for it. It's actually free. Study Manager is. Now, the product isn't, of course. You have to purchase that, but the software definitely is.
Wonderful, thank you. Our next audience member wants to know, we are getting a 6545XT QTOF system here shortly. Is the Study Manager available for our system?
Yeah, that's exciting to see. The QTOFs are great systems there. Unfortunately, Study Manager is not set up for the QTOFs at this time, the TOF, QTOF, or the single quads as well. Historically, those other instruments have not been pushed into the routine analysis side as much as the triple quads have. So unfortunately, Study Manager has been developed just for the triple quads and not for the TOF, QTOF, or the single quad line. There are other opportunities, though, if you were to look for things that are similar to Study Manager for high throughput or for like a walk-up-esque type of thing that are available on those products. But I would ask you to contact your Agilent rep to get some details about some of those other products that would help you with more of a walk-up or a high throughput software for your other instruments. Yeah, good question, though.
Thank you. And again, thank you, audience members, for your great questions that are coming in. Ryan, what traceability features are there for the users?
So that's a little bit of a broad question. I'm going to hope that I capture it correctly. So in traceability, I assume that it stems from how to make sure that the users are submitting it and everything's kind of tracked appropriately. As I mentioned in the demonstration that we did there, you can have it set up as like an SOP inside of your lab that would have individuals log in as, I think in the example I just used, operator, but you could have them actually have some sort of identifier that you could use. That's one way of addressing the traceability as far as making sure that the individuals that have submitted the samples, you can account to that. Another way is you can actually have it configured with your IT group to use your NT or your workstation login, and it can be coupled with that so that you would have additional traceability associated with it as well. Hopefully I captured that question correctly. Please extend that out if there's more to that.
Thank you. And Ryan, is the Study Manager compatible with LIS middleware?
Oh, yes, of course. So yeah, actually, a lot of the LIMS systems and the LIS systems will frequently use middleware, particularly the LIS systems. They'll obviously, the vendors of some of the major LIS softwares can't accommodate every vendor, so they'll go through third-party vendors or middleware that will do that. And yes, actually, we have quite a bit of interface in that. So one of the bigger ones, I think Data Innovations came up with a driver that they utilize for it there, so it's definitely compatible. But the other thing that works really well is almost every LIS or LIMS system has some way to accept either an SQL interface or a CSV, a basic CSV file. They can move those around, and the Study Manager program can use both of those. And again, if we worked with that particular middleware vendor in the past, as we have with like Data Innovations and a few other ones that unfortunately slipped my mind at this particular time, it's possible they may actually have the XML interface as well. So there is an incredibly high, I have yet to run into any middleware or LIS software that wasn't able to interface with the system in one way, shape, or form. Yeah, good question.
Thank you again. And we have time for one more question. Ryan, an audience member wants to know, do I have to use the automated data analysis?
No, you don't. And actually, this comes up pretty regularly. So the automated data analysis and the automated reporting has a lot of application for things that are kind of the set it and forget it type of assays. We see this sometimes in some screening assays and things of that nature, and they work really well for that. But there are others that do require a little bit more of a trained eye in the data analysis and the reporting, and they still want to be able to benefit from the batch Q sample submission, but they may not want to use the automated data analysis or reporting. That's okay. You can absolutely do that. You don't have to utilize the automated data analysis or the reporting. You could just set it up for just kind of a walk-up type of system, and that would absolutely work. There's no no conflict with that.
Thank you, Ryan, and thank you for your presentation today. Did you want to provide any closing remarks for our audience?
Yeah, actually, well, first and foremost, we called this venue the Healthcare Heroes, and I want to say thank you to everybody that's attended, and just thank you to all the healthcare workers out there in general. We really appreciate it in this tough time. Thank you for joining us, and thank you for listening to our presentations today, checking out our booths. Just really happy to see the attendance and the people, and just very grateful for all the work that's being done out there. So I'll leave it at that.
Thank you so much, Ryan, for your time today and for your important research. And before we go, I'd like to thank the audience for joining us for today and their interesting questions. And thank you again to Agilent for sponsoring these webinars. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of the registration. This webcast can be viewed on demand through May of 2021. Until next time, take care and have a great day. Bye-bye.